Hi students, this is your instructor Mr. Durst talking, and we are going to review the hierarchy of skeletal muscles. So this is kind of a review presentation just to help reiterate a lot of what we've talked about already. So as we look at a muscle, we'll start with the bone for a skeletal muscle. And then from the bone, we're going to go to the tendon, which attaches bones to muscles. The tendon will form and spread out into an fascia. The fascia merges with a membrane on the outside known as the epimysium, and the epimysium is going to have a bunch of bundles on the inside. One of the individual bundles inside of an epimysium is known as a fascicle, and the fascicle has a membrane around it known as the perimysium. You can think of a fascicle as a bunch of individual muscle fibers bundled by the perimysium. So we have, here we go. We have the one perimysium around the fascicle, and within the fascicle, we have many individual muscle fibers. Within one individual muscle fiber, we're going to have the cell membrane known as the sarcolemma. And then within the cell membrane, we have a bundle of myofibrils, or a whole bunch of individual myofibrils. And then if we zoom in on an individual myofibril, one individual myofibril is a bundle of myofilaments. So here we can see an individual myofibril, and then we have the thick and thin myofilaments within that myofibril. As we zoom in on regions or segments of the myofibril, let me clear the screen right here. And if we were to focus on these segments, let me clear that one more time. As we focus on these segments of the myofibrils from one point to the next point, one Z disc to the next C disc, we have the sarcomere. The sarcomere is the contractile unit of a skeletal muscle fiber. Within the sarcomere, we have the myofilaments, the thick and thin filaments. The thick filaments are shown in purple, made of primarily myosin, and the thin filaments are shown in pink and are made of primarily actin. So let's talk about our relationship between nerves and muscles. The muscle fibers, skeletal muscle fibers, are never going to contract unless we stimulate them with a nerve. And nerve stimulation is usually going to entail some kind of an electrical signal being sent to the muscle cell. Now, if we cut the nerve or we poison the nerve, for some reason, one of those two reasons, usually it's going to be severing, cutting the nerve, the muscle is going to be paralyzed. Paralyzation associated with severing the nerve or poisoning the nerve is known as a denervation paralysis. It can lead to denervation atrophy. Denervation atrophy is going to be shrinkage of the tissue. So trophy refers to the growth. A means without. So atrophy means shrinkage of the cells. The cells get smaller. And when we look at denervation atrophy, cells are getting smaller because they no longer have innervation. The innervation has been removed, D meaning without. So here's a photograph of somebody who has a spinal cord injury. Notice how the upper body of this individual is well developed. He has great skeletal muscle mass on his upper body. His upper arms, his chest has very well developed skeletal muscles. And look at his legs. His legs have very poorly developed skeletal muscles. He has denervation atrophy of his legs because he spends most of his time sitting in a wheelchair, not using his legs. As we're looking at the nerves that control a muscle fiber, that nerve is going to be made of individual neurons, and the axons of those individual neurons are going to travel from the spinal cord all the way to the skeletal muscle fiber. And I want to emphasize here that these somatic motor neurons can be quite long. They can go all the way from the brainstem or spinal cord to the individual skeletal muscles of the body. Somatic motor fibers are axons of the neurons, and the axons are going to go into the muscle directly. So on our screen right here, I should really switch colors. Let's go with orange. We have cell body of the neuron, the neurosoma represented by the circle, and the axon is represented by the long skinny line. And we have a green and a purple axon. The purple axon is going to innervate the individual purple muscle fibers. The purple dots represent individual purple muscle fibers controlled by that one purple axon. And then the green dots represent individual muscle fibers controlled by that individual green axon. This is important to emphasize. If we have one axon stimulated, one somatic motor fiber stimulated, all of the muscle fibers attached to that one motor fiber will contract. It's an all or nothing phenomenon. And if we look at one motor neuron and all the muscle fibers associated with it, we refer to that as a motor unit. That motor unit is the one fiber plus all the one nerve fiber plus all the muscle fibers that are connected together. And we can only contract whole motor units. This is the unit of contraction. So if we have really big motor units, we're going to have gross control of our muscles. If we have really small motor units, we're going to have very fine control of our skeletal muscles. All the muscle fibers of a motor unit contract in unison at the exact same time. And if we have a small motor unit, that's going to give us smaller or weaker contraction spread out over a wider area. Bigger motor units are going to give us more contraction spread out over a wide area. Why is it important to have motor units? Motor units allow us to contract our muscles for long periods of time. Think about the muscles associated with posture, or maybe you're carrying, I don't know, a gallon of milk from the grocery store and you're walking a couple blocks to take it home. You need to be able to contract your muscles for long periods of time. And to do that, oftentimes, we alternate one motor unit to another motor unit within the same muscle, maintain the same amount of tension within that muscle. One of the things that we notice is that as we fatigue our skeletal muscles, we are going to have kind of a quivering or a shaking of our skeletal muscles. And that quivering and shaking represents our body rapidly shifting from one motor unit to another motor unit very quickly, because those motor units are fatigued and running out of the ability to contract very quickly. So, as we look at a motor unit, the small motor units are typically going to be in our eye, hand, and facial muscles. And they're super tiny. We'll have only 3 to 6 muscle fibers per neuron. Large motor units, on the other hand, can have up to 1,000 muscle fibers per motor neuron. Now, these are the extremes. An average motor unit is going to have 200 individual muscle fibers. But when we talk about fine motor control, think of very small motor units, and then gross motor control is going to have the very large motor units associated with them. As our motor neuron sends its nerve fiber to the individual muscle fiber, we have something called the neuromuscular junction, physical interface between the nerve and the muscle. And where we have this interface, there's a little tiny space called synapse. And the synapse is going to be the physical space where the motor neuron and the muscle cell come in physical contact with each other. Now, as we're looking at the synapse of the neuromuscular junction, NMJ, the terminal branch of the nerve fiber is going to bulge out. And that bulging out is referred to as the axon terminal. As we look at the axon terminal, let me clear the screen here and shift colors. This bulging, or this axon terminal, is going to have storage vesicles filled with neurotransmitters. Because they're at the synapse, they're referred to as synaptic vesicles. And because this is a neuromuscular junction, the neurotransmitter in the synaptic vesicles is referred to as ACH, acetylcholine. And as that acetylcholine, ACH, is released, it needs to move physically across the synaptic cleft. That synaptic cleft is the physical space between the motor neuron and the muscle cell. So it's the gap of the synapse. And those neurotransmitters, the acetylcholine, will diffuse across the synaptic cleft to get to receptors. And there are receptors are these blue, kind of dark blue, almost purplish proteins that are represented here. Now, as we have this nerve impulse, this electrical signal, sometimes referred to as an action potential, travel down the motor neuron to the axon bulb or the axon terminal, the acetylcholine will be exositized from those synaptic vesicles. And the acetylcholine receptors on the muscle side or on the sarcolemma side are going to receive the acetylcholine. There are going to be folds in the sarcolemma directly beneath the axon terminal that house lots of acetylcholine receptors. These folds serve to increase surface area. And by increasing surface area, we can pack more acetylcholine receptors in a smaller space to better receive those acetylcholine molecules that are transmitting the signal from the nerve fiber to the muscle fiber. Now, if we don't have enough receptors, enough acetylcholine receptors, there's a disease associated with that known as myasthenia gravis. So if we look at somebody with that disease state, myasthenia gravis, it's typically going to be associated with the inappropriate number of receptors for acetylcholine, not an inappropriate amount of acetylcholine, and not an inappropriate amount of electrical activity of the motor neurons. That's all we have for this recording. As always, please feel free to shoot me an email if you have any questions or comments, or post them on the discussion board. Happy studies.